Hello dear student, myself Adke Asmita Adke. I am working as an assistant professor in Department of Chemistry from SVKT College, Deolali Camp, Nasik. Today we study about the role of Fe in metalloprotein. First of all, we understand what is bioinorganic chemistry. Bioinorganic chemistry is the branch of chemistry that focuses the role of inorganic element and compound in a biological system, how metal ions such as Fe, magnesium, copper and other interact with biomolecule like protein, enzyme to perform a vital function in a living organism. Metalloproteins are proteins that contain metal ion as a key component of their structure and function. These metal ions can be directly involved in biological activity of the protein. Here we consider the Fe. So, Fe play a crucial role in both metalloprotein and metalloenzyme as it is essential for their structure and function. It is important for oxygen transport, electron transfer and catalysis. Iron plays important role in hemoglobin. So, hemoglobin is a protein found in a red blood cells that it is responsible for carrying oxygen from the lungs to the rest of the body and returning carbon dioxide from the body back to the lungs for exhalation. It consists of 4 subunits containing an iron atom that binds to oxygen. This ability to bind oxygen is what allows hemoglobin to perform its crucial function in oxygen transport. Hemoglobin, it is a protein made up of 4 subunits containing hemigroup. The hemigroup contain 4 ferrine ring. Hemoglobin is a globular protein with molecular mass of 64 K Dalton. A healthy human has 12 to 20 gram of hemoglobin every 100 ml of blood. When oxygen binds to hemoglobin, it is called oxyhemoglobin. When oxygen not binds to hemoglobin, it is called deoxyhemoglobin. Function of hemoglobin. First, we know the important function of hemoglobin, that is oxygen transport. So, hemoglobin bound to oxygen in the lungs transport it through the tissue and organ throughout the body. Then carbon dioxide transportation. Hemoglobin also helps to carry carbon dioxide, a waste product of metabolism from the tissue, back to the lungs where it is exhaled. This is the structure of hemoglobin. Next, we discuss the oxyhemoglobin. It is a molecule that carries oxygen throughout the body. Fe present in +3 oxidation state. It is a low spin diamagnetic octahedral molecule, red in color. In this diagram, we see the Fe is present in center metal atom. In that 4 nitrogen attached from 4 ferrine ligand system and 5th coordinating site containing attachment of nitrogen atom from histidine amino acid, and to the 6th side of oxyhemoglobin O2 molecule is attached. Then we discuss the deoxyhemoglobin. It is also a 5 coordinated molecule in which Fe is present in +2 oxidation state. Fe is present in high spin paramagnetic state. In this diagram, we see the Fe lies 0.5 angstrom above plane containing 4 coordinated nitrogen atom of 4 ferrine ring and nitrogen atom of imidazole of histidine group. Then myoglobin, it is also a protein found in a muscle cell that binds to and store oxygen. It is also play crucial role in delivering oxygen to muscle tissue, especially during the different types of physical activity. Myoglobin is similar to hemoglobin, which carries oxygen in the blood, but it is specially adapted for oxygen storage and transport within muscles. Characteristics of myoglobin. Myoglobin, it is relatively small, single chain protein consisting of about a 150 amino acid with one hemigroup that binds to oxygen. Its primary role is to oxygen transport within muscle cell. During exercise, myoglobin releases oxygen when muscle demand for oxygen increases. Myoglobin has a high affinity for oxygen in the lungs and release it to muscle cell when needed. This is the structure of myoglobin. In this diagram, we see the myoglobin is a small protein of molecular weight of 17,000. It consists of one polypeptide chain with hemigroup, and the peptide chain consists of 150 to 160 amino acid, and the Fe atom consists at the center of hemi is bounded by 4 porphyrin nitrogen and 1 nitrogen atom from imidazole side chain of histidine. Next, it is a hemanthrene. Hemanthrene is another class of oxygen transport molecule in a several organisms. It is present in some aquatic life, marine worms. In hemanthrene, hemigroup is absent. It is a dimer of 2 ion atom, has important function of oxygen transportation, respiration in case of several marine worms. The hemanthrene which is not taken up oxygen is called deoxyhemanthrene. The hemanthrene which has taken up oxygen is called oxyhemanthrene. Function of deoxyhemanthrene. It is the main function is bind and store oxygen. It is play crucial role in the transport and regulation of oxygen in organism tissue, and it is colorless. It is also known for its ability to reversibly bind to oxygen through, and it has a lower oxygen affinity than hemoglobin. Function of oxyhemanthrene. It is a primary function is to transport and store oxygen. Similar to vertebrates, the protein is able to bind oxygen molecule due to the presence of ion in structure which forms an oxygen binding complex. When oxygen is present in oxyhemanthrene, picks it up and carried it through the organism body to tissue that require oxygen for metabolic process. Then we consider the ion in ferritin and transferritin. Ferritin and transferritin are two important proteins involved in iron metabolism in the body, playing distinct role in Fe storage and transport. Ferritin is the primary protein used by the body to store iron. It is found in all cells but primarily in liver, spleen, bone marrow, and it is stored in iron in non-toxic form. In this diagram, we see the structure of ferritin. It is consists of hollow sphere in which Fe3 stored in a crystalline material. The hollow sphere is surrounded by protein called apoprotein. The apoprotein consists of 24 peptide chain. Subunit has a molecular weight 18,500. The structure of ferritin allows it to hold up 4,500 atoms in its core. Then this is the structure of transferritin. In this structure, we see the Fe is present in +3 oxidation state. Then carboxylic group from aspartic acid is present, carbonate group also, and histidine chain. The regulation of ion mobilization and storage is done by transferritin and ferritin in mammals. Now we will consider the mobilization, that is transportation of ion in mammals including human. This main function done by transferrin. The ion absorbed from the food must be transported to body through function by protein of the transferrin like glycoprotein. This glycoprotein has molar mass 80 kilodalton that tightly bounds to Fe3 ion. The structure is mostly distorted octahedral type structure was found in the 6 coordinating sites of ferritin. The coordination sphere consists of 2 phenolate oxygen from tyrosine, 1 imidazole nitrogen from histidine amino acid, 1 carboxylate oxygen found in Fe3 of transferrin from aspartic acid, and 2 oxygen from carbonate group of amino acid. Next, we discuss about the iron sulfur cluster. The iron sulfur cluster has an important function as a electron carriers in a biological redox reaction such as photosynthesis, nitrogen fixation. Then iron sulfur protein are classified as a rubedoxine. Here we first consider the what is rubedoxine. Rubedoxine, these are found in anaerobic bacteria. These are very small FES protein which have low molecular weight 6,000. These protein contain 50 amino acid and single Fe atom is in oxidation state. It is bound to 4 sulfur atom. The sulfur atom attached to Fe atom from amino acid cysteine which is linked for protein chain. This is the structure of rubedoxine. In this structure we see the iron is present in center in +3 oxidation state and 4 sulfur are present in. Then it play a key role in electron transfer in various biological process. The function of electron transfer in metabolic pathways such as nitrogen fixation and sulfur metabolism. Next type of ferritoxin. Ferritoxin, these are several types of ferritoxin, that is one contain 2 FAS cluster, second contain 4 FAS cluster and third contain 8 FES cluster. These group are generally bound to sulfur atom from cysteine amino acid. The ferritoxin are generally occur in plants, bacteria and animals. Ferritoxin are a group of iron sulfur protein that are found in various organisms including bacteria, plant and animal. They typically consist of a small protein with a molecular weight around 10 to 12 kilodalton. The structure of ferritoxin is characterized by the presence of iron sulfur cluster, and these clusters are active sites of ferritoxin and play a key role in electron transfer. Here we see the function of ferritoxin. First function is electron transfer. This is the primary function of ferritoxin, and that is transfer electron in a various biochemical pathway, and this electron transfer occur through iron sulfur cluster which can change oxidation state of ferritoxin to participate in redox reaction. Second function is photosynthesis. In plants and cyanobacteria, ferritoxin are essential. They are involved in the transfer of electron from photosystem 1 to NADP plus to form NADPH, which is required for the photosynthesis. Thank you.